Welcome to the State Forestry Research Center Eberswalde, Landeskompetenzzentrum Forst Eberswalde in German, or LFE for short. Here, scientists conduct application-oriented research and knowledge transfer on forest ecosystems in the northeast German lowlands, providing a wide range of scientific services for the state forest, as well as for private and municipal forest owners. The tasks include forest inventories and monitoring programs, the supervision and evaluation of long-term experiments, silvicultural and forest growth experiments, as well as research activities on forest protection, forest ecology, forest genetics, and wildlife ecology. This is the place where Eric analyzes the leaf samples that he collected in Demmin. In order for the leaf constituents not to deteriorate, the samples were frozen on dry ice in the field and quickly transferred to the freezer in the lab. Now Eric collects the samples out of the freezer. It's minus 80 degrees Celsius, and Eric needs to be careful not to collect frostbites as well. That's what the gloves are for. To measure the leaf constituents, the samples are milled to powder. To do this, Eric transfers the samples from the tubes into grinding containers. They are made from stainless steel and contain a stainless steel ball to grind the samples by impact movements. He carefully places the grinding containers with the samples in a polystyrene box and fills it with liquid nitrogen. Then he places a glass plate over the box so that the liquid nitrogen does not splash out. That way, the samples are shock frozen for 3 minutes at approximately minus 200 degrees Celsius. After 3 minutes, Eric removes the grinding containers from the liquid nitrogen and tips the remaining nitrogen back into the Dewar flask. Now, the grinding containers are clamped into a beater mill and ground for 3 minutes at 20 beats per second. Once the samples are ground, Eric removes the grinding containers from the beater mill, opens the containers, and transfers the samples to Petri dishes using a spatula. The Petri dishes are sealed with parafilm. And again, the samples go into a freezer, again at minus 80 degrees Celsius this time. Next, the samples are dehydrated in a freeze dryer. This fancy device removes the moisture from the samples with a negative pressure of approximately 20 Pascal. The moisture is collected and frozen by a minus 80 degrees Celsius cold trap under the sample. Once the samples are dry, Eric releases the pressure and stores them in a desiccator to ensure that the samples are protected from moisture and light. The samples are weighed and the leaf constituents are extracted. The extracts, a clear liquid, of the samples are measured in a spectrophotometer for proteins, chlorophyll A and B, carotenes, carbohydrates, starch, certain phenolic groups, and ascorbic acid, among others. Meanwhile, Micha and Pia meet Ralf Ketzel, the head of department, next door to discuss recent results. Climate change is increasing the demand for reliable information on forest health. Yet, due to a lack of precise data, many current assessments remain contradictory and speculative. Forest remote sensing holds great potential to complement terrestrial forest condition surveys. However, uncertainties remain, particularly in the spectral differentiation of tree species, because clear links between spectral signals and tree physiological or chemical traits have not yet been firmly established. This is especially true for mature trees, where data is difficult to collect under natural conditions. As a result, we lack a solid understanding of how signals measured above the canopy relate to internal tree stress indicators, known as biochemical biomarkers. This gap between remotely sensed data and the physiological and biochemical processes within trees is exactly what the FEMOFIS project seeks to close. On the one hand, the laboratory in Eberswalde analyzes biochemical indicators for tree vitality and stress. On the other hand, the project members, for example from the Technical University of Berlin, investigate which of these indicators can be recorded using hyperspectral remote sensing. Therefore, team members acquire measurements from multispectral, hyperspectral, and thermal sensors at various levels of detail. From very precise measurements directly on the leaves to spatially accurate drone images to aircraft and satellite images, as well as terrestrial laser scans. For example, these measurements allow the assessment of leaf water and chlorophyll content and biomass, as well as the determination of crown size and tree height. As the project progresses, we will see what other vitality indicators can be remotely detected. These remote sensing data are combined with regular measurements of microclimate, precipitation and soil moisture at several depths, sap flow, root and stem growth, photosynthetic activity, and litter fall. In addition, leaf constituents like chlorophyll, nitrogen, water, and protein content are analyzed in the lab. Trees have different means to deal with unfavorable conditions. For example, they defend themselves by producing chemical defense substances that ward off insects and pathogens. They also form thick bark and resins as physical barriers. In the northeast of Germany, with its light, sandy soil and, especially in recent years, often poor precipitation, the trees are suffering from water stress. Interestingly, even neighboring trees can choose very different strategies to deal with this stress, almost like humans. Their strategies affect photosynthesis, sap flow, growth, storage, and defense. Based on their detailed measurements, the FEMOFIS team could identify differing reactions of trees to environmental conditions. Go ahead and check out a selection of results on specifically interesting tree individuals on the FEMOFIS website yourself.